So I'm Heather Dewey Hagborg, and I'm an artist, and I'm a student at Rensselaer Polytechnic Institute right now in upstate New York. Some alums in the audience. And I'm going to just talk to you today about a project I've been working on for the past year and a half called Stranger Visions. And it's a work in progress, so keep that in mind. So in April, I received a rather unusual package in the mail. It came from a medical examiner by the name of Hal Brown in Wilmington, Delaware. And he was sending me this package after several very lengthy telephone conversations. He told me he was sending it, and he told me what was in it. But when it arrived, I had a really hard time opening it. It sat on my desk for several days, filling me with this strange mixture of excitement and dread. Because I knew that what was sitting in this nondescript box was a human rib. So how did a PhD student in electronic arts end up receiving a human rib in the mail? It's kind of a strange story, and strangely enough, it begins with single hair. So I was actually sitting in a therapy session, not in Freud's office, but in a different office, and I was staring at a painting on the wall, and I noticed that the glass covering the painting had a crack in it, and in that crack was lodged a single hair. And as I sat there, with the purpose of thinking about myself, instead I just started focusing on this hair and trying to imagine whose hair it could be, what they might look like, what they might act like, how crazy they might be, and how they came to leave this hair in this space. And it was really that obsession with thinking about what I could know about someone from a single hair that led me on this kind of strange journey. So I'm a research-based artist, and my work generally tends to begin with some kind of question. So things that I've thought about in the past are things like, can a computer be creative in the way that we think about a human as being creative? Or if a facial recognition system dreamed, what would it dream about? So these are some faces from some of those early experiments in hacking facial recognition algorithms and taking them and getting them to sort of turn back on themselves and generate faces instead of recognizing faces. So most recently, I've become fascinated with this question of what I can know about someone from the traces that they leave behind, from discarded genetic material like a hair. So I started really focusing on DNA that we leave behind. And the funny thing is that once you start thinking about it, you start noticing evidence everywhere. So public bathrooms, the sidewalk, a bar, you leave your glass on the bar, the subway. People are leaving their DNA around all the time without ever thinking about it. And it struck me here that the very things that make us human, hair, skin, saliva, become a liability as we constantly face this possibility of shedding this material in public and leaving artifacts which anyone could come along and mine for information. So coming from a background in electronic arts, I'd already thought a lot about surveillance and privacy online, like I'm sure many of you have had probably one too many conversations about Google and Facebook and oversharing and all of this. And I'd thought a lot about surveillance on the streets in terms of things like facial recognition and wiretapping and speech recognition as well. And I'd experimented a lot with these things in my work. But I'd never really considered this emerging possibility of genetic surveillance. I think I assumed going into this project that that was still in the realm of science fiction. But I wanted to find out for sure. So I signed up for what's called a biotechnology crash course at GenSpace. And I don't know, how many of you know GenSpace? I can't actually see you, so. It's a do-it-yourself biology lab in downtown Brooklyn. For those of you who are New Yorkers, I highly recommend that you check it out. It's a really fascinating place filled with interesting people. So I signed up for this biotech crash course, and I learned how to extract DNA and how to amplify certain regions of it using a technique called PCR, polymerase chain reaction. And so basically what PCR is it's a series of steps where you heat and cool purified DNA along with what are called primers, these specific chemicals. And this very programmed series of steps allows that DNA to multiply. So you are amplifying little sections of DNA. And if you then send those little sections to get sequenced, you can analyze what comes back to look for specific traits. So I started digging into this research to try to answer these questions, and I discovered that there's actually a whole lot that an amateur like me can find out about a stranger from a random forensic sample, relatively cheaply and relatively easily given enough time. So for example, by amplifying certain regions of your mitochondrial DNA, this is the DNA that is passed down directly from your mother. It's not DNA that codes for any specific traits. It's kind of this residual DNA. So by amplifying certain regions of your mitochondrial DNA, I can make a pretty good guess as to where your maternal ancestors were hanging out about 500 years ago. This is before the era of intercontinental travel. And it's classified into these categories called haplogroups, and you can see these letters represent these haplogroups here on the map. It's not a perfect system, but it gives a pretty good estimate. So for example, my maternal ancestors are of the group U, and you can see they're up there in Northern Europe. By examining a section of chromosome 1, I can tell if you're likely to have malaria resistance. By looking at chromosomes 14 and 15, I can tell how likely you are to have blue eyes, blonde hair, and pale skin like me. So all of these little base pairs, which are called SNPs, single nucleotide polymorphisms, give me clues as to what you might look like. So if you can see in the sequences up here, there's one sequence of DNA on top, one on the bottom, and they vary only in this one particular base pair, AT on top, CG on the bottom. So that's a SNP, right? A single place where something varies. And it's not the same as a gene, right? So genes actually code for traits. They actually are involved in the creation, in the expression of these traits in your body. SNPs are correlations. So they're things that are generally found in what are called genome-wide association studies, which are these big statistical studies where you take lots and lots of genomes and lots of information about a person, and you find correlations. So I wanted to do something with this. As I'm starting to dig into it, starting to understand how things work, I wanted to do something artistic with this to call attention to the questions I had about this technology and these emerging issues. But I didn't want to do this. Right? The caption here is, "Immortalize your Valentine as a piece of art." It's what I didn't want to do. I wanted to do something that people would have a visceral reaction to, that you'd walk up to and say, wow. And as you've seen, I had worked with faces in the past, and I was really struck by how they act as indexes to even more intimate information due to their associations with identity. So this led me to thinking about the form of the portrait and thinking that by representing what I knew about what someone looked like, it would be a way of pointing to these deeper questions of genetic privacy without actually doing the potentially evil things that I could imagine, right? I didn't want to actually try to figure out who people were from their DNA or post their entire genome online or analyze their disease risks, right? Just a few of the things that I can imagine one could do. But I wanted to point towards the fact that that was becoming possible. So I created this master list of about 50 traits, and these are all from available published research. This is not anything new. What I'm doing here is really taking these pieces and putting them together. So in addition to ancestry information, I have 50 traits, and most of these are accessible from 23andMe, and some of you might have done 23andMe tests, or you might be familiar with it. It's a personal genomic profiling service, and how it works is you go to the 23andme.com, you pay $100, and they send you a kit, you spit in it, send it back, And then they give you this very comprehensive profile of their best guesses as to your ancestry, your disease risks, and your traits. And the traits are kind of the fun part there, but they were the part that I was the most interested in. So, I dug into that, and there's also a site called Snipedia, so kind of like Wikipedia for SNPs, and started pulling out all these different traits that I thought related to physical appearance. And then I started going down this list in the lab and just trying to analyze them. And I'm going to show you, I have a little video here that I'm going to narrate over, which describes the process that I've gone through in this piece and shows some of the result as well. So the first step is looking for samples. And I tend to look for samples just in my daily routines, as I'm going about my business, traveling into the city. And so if you're doing this, you want to look for things that look relatively fresh, right? I tend to collect hairs, cigarette butts, chewing gum, and fingernails. And you want to look for things that look like they've been dropped or shed relatively recently, because the older a sample is, the harder it is to extract DNA from it. So there's lots of good spots in the city for finding these things. And so this is one of the labs that I work in. It's GenSpace in Brooklyn, which I mentioned earlier. I also work at a lab at Rensselaer, upstate New York. And so I take the samples into the lab, hair, for example, and I'll cut off a tiny piece of the sample. And then I'll cut the tiny piece into smaller pieces and add a series of chemicals and walk through a series of steps in a protocol, very much like a recipe, like I was baking a cake. And take this mixture, put it in this microtube, and shake it up and heat it and centrifuge it. And hopefully at the end of that process, what I'll end up with is purified DNA. If I successfully get the DNA from the sample, then the next thing I do is I walk through a series of PCR reactions, what I was explaining earlier, right? So the PCR it's kind of like a computer that just heats and cools according to a program that you enter into it. And so I just walk through these steps, and then hopefully at the end of that process, I'll run this gel electrophoresis, and hopefully I'll see bands where I hope to see them showing that I successfully amplified this particular region. So in this example, you can see that there's two samples here and that they are both successfully amplified. I'm just going to try to pause this for 1 second. And so I'll take the result of this PCR reaction and send it out for sequencing. And what I get back are basically text files. They're just text files filled with ATCs and Gs. I mean, it's kind of surprising just how simple that is. And then I go through a bioinformatics process where I line up these files and I look at that particular SNP, right? That one area that I was interested in that codes for a trait, and pull out all of these traits. So for one sample, I'll look at a series of these different traits, along with ancestry information from mitochondrial DNA, which is a little bit more complicated, but similar idea. And then these get fed into the software that I'm working on. So the software is built off of a project from a group in Basel, Switzerland, called the Basel Morphable Model. And this is a 3D model of a face that you can parameterize in terms of things like age and gender and weight. And I basically extended this model so that you could also parameterize it in terms of ethnicity and specific traits like lighter or darker skin and eye color. And the morphable model is totally open source too, so any of you could request access to it and download it and play around with it. So from that 3D model that's generated, I solidify it in Blender or Maya, and then export it and send it to the 3D printer. And so this is an early experiment with the 3D printer. And here's one of the faces coming out of the powder in full color. And then, because it's very fragile in that state, being infiltrated with this kind of super glue-like liquid. And then, of course, the final results are exhibited in a gallery and online, along with the samples that I collected them from and documentation about where they were found and what traits I know about that person from that DNA. Okay, so the first question I always get in this project is about accuracy. I started this project actually with a self-portrait. So I started this when I was doing a residency at IBeam, and it took me a really long time. It took me a lot of attempts and failures to get anywhere with this project. But the first kind of proof of concept that I did was a self-portrait. And what I did was I took my profile information from 23andMe and fed all of that information into this software that I was developing. So this is containing all of the traits that I was interested in, right? This is like my dream data. And so it's interesting, so this is the self-portrait. What's really interesting about exhibiting this self-portrait is that half of the people who see it say, wow, that looks nothing like you. And the other half say, wow, that looks just like you. So the truth is probably somewhere in the middle, right? The portrait, it doesn't look like me in the way that you would think about, you know, your friend and who looks like your friend. But we have certain things in common. And that's kind of where this technology is at right now. You can create a kind of general likeness or a portrait that has certain family resemblances to the person themselves. But it's not going to be a reconstruction. It's not going to look just like this person. And furthermore, you know, this is kind of best case scenario. It can be even further off if you have particular traits that science hasn't even looked at yet, right? Like if you happen to have a really narrow chin, for example, there's really no data yet on how that is connected to genetics. So the other portraits that I've worked on so far are all portraits of strangers. So these are things where I find a sample on the street and then extract the DNA and analyze it. So I found this cigarette butt on the sidewalk right by my apartment in Brooklyn. And after extracting the DNA and analyzing it in the lab, I discovered that it is from a person of Eastern European descent. It's a male with brown eyes. More recently, I've discovered that he also has an average tendency towards being overweight and a slightly larger than average nose. There's a lot more that I hope to discover about this person through continuing in my lab work. This is a preliminary, in-progress portrait. All of the portraits that I've done, I consider to be in progress. And as I continue to work on this in the lab, I plan to analyze more details of his ancestry, his skin complexion, right? Whether he has lighter or darker skin, whether he has freckles, his hair color, and other dimensions of his face, such as will his eyes tend to be closer together or further apart. This is a female of European descent, a male of South American or Native American descent, and another male of West African descent. This one is from a cigarette butt I actually found right on the sidewalk outside of GenSpace. So there's been a lot of confusion, I think, in the media about what this project is and what it isn't. So it isn't a scientific discovery about how to reconstruct faces from DNA. It's definitely not that. These are not reconstructions. These are more like sketches, right? They're using my subjectivity and the interpretation and speculations of scientific research and combining these things. So this isn't a dazzling new technology, and this isn't a startup. I'm using available research and tools. This piece is meant as a provocation in the spirit of tactical media. It's supposed to make you think about privacy, about genetics, and about our biotechnological future. The point isn't that I know everything about a person from a piece of chewing gum, but rather that I, as this amateur, can know as much as I do and can potentially learn a whole lot more. So, returning now to the package. This art piece got covered in a forensic magazine, and I started getting emails from the strangest people. The Moscow police, all kinds of forensic startups. And so all of these things I turned down for both practical and ethical reasons. And then I got this email from Hal Brown. Your amazing project could help identify a murdered woman. Don't get emails like that very often. And Hal said, my goal is to identify these poor women, to give them back their identities and send them home to their families. Most are historical cases. Some have been unidentified for decades. Wow. I was hesitant to work with the police on suspect cases, but this seemed like a genuine possibility to do some good without the same risks that one would take on when looking for a suspect. So I was intrigued, and we continued to talk. And Hal began to tell me the details of this case, the first case. A woman was found November 25th in 1993 in an overgrown field in William J. Baxter Memorial Park in Odessa, Delaware. She was found by a retired Wilmington PD cop who was hunting in the area. Her body was positioned in a west-east direction with her head facing west, her feet facing east. No jewelry or clothing was found on the body, and it appeared that she'd been there for 3 months. The more I thought about it, the more I felt like I could contribute at least something to this case. Not necessarily to create the most accurate portrait based just on the DNA, but that possibly by adding in some genetic details to what they already knew, we could make a better portrait. So she's remained unidentified since 1993, despite many attempts to create her likeness. And so what we know is that she was between 20 and 25, possibly of Asian American descent, female with brown hair, about 5 feet tall, with an overbite, a distinctly forward chin, and high cheekbones. So there's a lot that we can know from the forensic remains, from the bones, but there's plenty that isn't known immediately as well. So whether she tends to be overweight or not, what her nose looked like, what eye color she had, what her skin tone was. So there's plenty of genetic factors that I think could be useful in creating a more accurate likeness, or even a series of different probable likenesses, right? You could imagine how you could have 3 different portraits that you generate from the combination of her bones, her skull, and the genetic information. So the genetic portrait here becomes like one more tool in the box, something to add to the information we already have, building on the information in the bones. And so I thought this all through, and I decided I would try to help. And then the package arrived with the unidentified rib. And there was a new kind of gravity to this work, a new kind of responsibility came with this. And suddenly this project went from being an attempt at critique to becoming an opportunity to really do something real, something that could help someone. And after the box sat on my desk for several days, I did eventually open it with a couple of my professors at Rensselaer, and we did it together. We opened it up. We went through the DNA extraction protocol. It was a very intense and emotional moment for all of us. But the preliminary tests looked successful. It looked like we did successfully extract the DNA. So next time you talk to me, I may have some more clues as to what she looked like. Hopefully. So what are the implications of this work? This project has primarily raised questions for me. What are the implications for personal privacy if a government, corporation, or private individual like me can read your DNA? How will it affect social and political interpretations of law enforcement, for example, if police analyze a hair from a crime scene and discover that someone's of a particular ethnicity? How might that have impacted the Boston bombing case, for example? So will this technology simply reinforce cultural stereotypes that are already so problematic and become a new form of racial profiling? That's one of my primary concerns. Now, interestingly, this issue already came up in the Netherlands. As I was doing research early on in this project, I came across an article in Science describing the story of a young woman who was brutally raped and murdered. And after a year, the police still had no suspects, and the rural community in which she had lived was beginning to point fingers at the local Middle Eastern population, something familiar to us here in the United States. And despite the illegality of genetic profiling in the Netherlands at that time, a forensic biologist working on the case decided to go ahead and extract DNA from a semen sample anyway. And what he discovered was that it was left by someone of European descent. And this was interesting, and this, in this case, actually helped to calm the ethnic tensions that were swelling in this area of the world. But you have to wonder what would have happened if the result had been just the opposite. What kind of reaction from the community could that have engendered? So the Dutch passed a law regulating this practice, which is called forensic DNA phenotyping. This is kind of the official term for what I'm doing. And the law that they arrived at allows phenotyping for facial characteristics for things like eye color and ethnicity, but prevents analyzing for disease risks or things that they consider to be invisible. So the logic here, I think, is twofold. On the one hand, that DNA can show us what a witness might tell us, and on the other hand, that we want to try to avoid disclosing potentially very sensitive information publicly and even possibly to the individual themselves. So that's one of the privacy concerns associated with forensic DNA phenotyping. So for me, this kind of raises this question of how does forensic phenotyping compare, for example, to the validity of an eyewitness, another thing that's come under a lot of scrutiny and criticism lately. And also, how accurate does the technology need to be used in the courtroom? And I think it's important to learn from history here, history of similar technologies. So DNA typing or profiling, we're all kind of used to seeing these DNA fingerprint images from TV. So DNA typing was introduced to the courts with very, very little regulation. It was essentially discovered and then introduced to the courts immediately. And there was a lot of corporate interest involved. And this essentially left it up to the lawyers to challenge the technology. And so what happened was Barry Scheck and Peter Newfield, who went on to form the Innocence Project, which many of you I'm sure know, did exactly this. They questioned the technology that everyone else, even the scientists, were accepting point blank. And eventually this led to these National Research Council reports in 1992 and 1996, which formalized protocols and standards regulating this new DNA technology. But it was a really long process to get there, and they had to do a lot of fighting. But their passion ended up in a better and more accurate technology. So I think perhaps we can learn from this, and it would be advantageous as we're in the infancy of thinking about this new technology to think very carefully about how we implement them, to proceed more cautiously and more judiciously than we did with DNA typing, and to avoid some of the things that we know right off the bat are going to be problematic. Questions about accuracy, contamination, standardizing protocols, and the limitations, really outlining what are the limits of this technology, what can we know and what can't we know, and how do we explain that to the people who are going to use it. So in the United States, this practice, DNA phenotyping, is currently completely unregulated at the federal level, though there are a few states that ban it entirely: Indiana, Rhode Island, and Wyoming. And also, how accurate does the technology need to be used in the courtroom? So for me, this kind of raises this question of how does forensic DNA phenotyping compare, for example, to the validity of an eyewitness, another thing that's come under scrutiny and criticism lately. DNA databases are expanding every day. Some require that even someone convicted of a small misdemeanor submit DNA to the database, and as many of you probably saw in the recent Supreme Court ruling, if you're even accused of a crime, you can be forced to submit your DNA to the database. And what's problematic about this is who is part of these populations that are being subjected to the database, right? I mean, this is a racially skewed database, and this is then the database that is called upon when there is a kind of a cold case. This is the database that we draw upon when we look for suspects. So it's very problematic. Also in the past year, a kit has been released called H-IRIS-Plex, and this allows the police to tell from a forensic sample at a crime scene what a suspect's hair and eye color are likely to be. And there's also a company called Sorensen Forensics that allows the police to send in a forensic sample for ancestry or ethnic profiling. So the police are doing this. They're doing it now. And even if they don't have a fancy software that takes all this information and generates a portrait, they're at least taking this information and using it to inform how they look for suspects and probably how they create sketches of suspects as well. And the thing is that phenotyping isn't like DNA fingerprinting. DNA fingerprinting came with a whole bunch of issues of its own, but phenotyping is even more probabilistic. So there's a chance of a certain strength that I'll look a certain way from my DNA, but it's almost never a certainty. So this is my global similarity chart from my own 23andMe profile. And if you look at this, I could look all kinds of ways based on these bar graphs, right? The biggest one is in Northern Europe, but there's a pretty big one in South America too. So I think the problem here is that while the science behind this technology is probabilistic, we have this idea that it's written in our DNA. And this gives it an aura of objectivity that I think we need to be really careful about. So conclusions. I don't really have too many conclusions yet because this project is still continuing to grow and evolve. And a lot of these policy questions are just really starting to clarify for me out of this gray, murky mass that is the law regulating genetic and genomic issues. But one of the things that I'm most excited about right now is a series of policy conversations that I'm starting to have about the issues raised by this work. So this began in DC last Monday, and I met with a professor of law there and a scientist. And we had this very provocative conversation. It was really interesting, and I learned a lot about what the law does and doesn't say, and how actually the majority of it is in this very ambiguous zone that's still being defined. And it's really waiting for a precedent, or perhaps instigations like these, to help it become defined. So in these conversations, we discussed different laws in different states, questions of consent and anonymity, and how those shaped the impact of these laws, and also the idea of surreptitiousness, right? Because there's several states that have laws that regulate the surreptitious collecting of DNA with the intent of preventing someone from, for example, taking your cigarette butt and trying to determine if you're the father of their child. So there are all of these kind of interesting laws that are different state to state and that have really yet to be challenged or really even used. So there's a lot of legal questions about the work that I'm doing, and depending on the country or the state that I'm in, the legality varies. So a simple example of this is actually 23andMe. I don't know how many of you might be from New York and have done 23andMe kits, but New York has a law against sort of direct-to-consumer genomic profiling. So you're supposed to go through your doctor, basically, to get any kind of genetic tests. So in order to do your 23andMe kit, you have to get on the PATH train and go to New Jersey and pop it in the mail there. So it's interesting to see sort of how the laws roll out and also how we work around them. And this series is continuing, and I really want to invite you all to participate in this. So the series continues this Thursday at Gen Space in downtown Brooklyn. If you happen to be in New York, I invite you to come. Hopefully someone from the ACLU will be there, and hopefully someone from the FBI will be there as well. So that'll be interesting. I'll also be showing the work itself in person. And so maybe the best conclusion I have to offer right here is really an invitation to invite you to continue this dialogue with me in person at these kinds of events, as well as online. But I just want to finish by encouraging you to get involved. If you think privacy matters, if you're concerned about anonymity at all, do something about it, because we may well be the last generation that even notices what we're losing. Thank you.